Hello, Assalamu alaikum. In this video, we will understand about the anti-diabetic effect of silymarin compounds. So, collectively, we will understand the silymarin, so how it is anti-diabetic, and let's understand about that all things. So, remember, this is the milk thistle, is the plant which that contain this compound. Whenever you will eat, the carbohydrates go into the blood circulatory system is known as absorption after the digestion. This glucose will rush into blood is the hyperglycemic effect will release the insulin from the beta cell of pancreas. This insulin spike by beta cell and it will target to the several tissue like myocytes, adipocyte and liver as well as other cell. So insulin receptor is important. Remember, for type 2 diabetes mellitus, the insulin receptor will damage, while the type 1 diabetes mellitus is the beta cell will be damaged. This insulin receptor will allow to glucose inside through the GLUT4 channel facilitate. So this is the facilitate diffusion. The glucose will enter into the cell, increase the glucose for the normal level of the glucose in the blood. Glucose will lead to catabolize to produce pyruvate and ATP. This is the glucose will enter when into the beta cell when rise in the glucose in blood circulatory system. This increase of glucose will lead to influx of the calcium also to trigger the insulin vesicle to exocytose. But in the pancreas, the inflammation will lead to cause the beta cell destruction. This inflammation we will all things will understand through the milk thistle how it is helpful. So beta cell destruction will lead to hyperglycemia, ultimately lead to hypertension and other complications. So let's understand about the inflammation and oxidative stress due to the pollution, smoking, drug and medication will lead to reactive oxygen species increase, lead to cause oxidative stress. Silymarin will inhibit this effect and their target, the silymarin target is several. The oxidative stress and reactive oxygen species will lead to produce a necrotic factor kappa B, necrosis factor kappa B. This necrosis factor kappa B will trigger due to the IKK alpha, beta and gamma subunit of the protein. And this trigger and also this will lead to endoplasmic reticulum stress will lead to increase this necrosis factor kappa B, and this necrosis factor kappa B will trigger the interleukin 1 beta 6 and tumor necrosis factor alpha. This tumor necrosis factor alpha and these complex cytokines will lead to activate the basically inflammation of the beta cell and destruction. This further on the insulin receptor substrate 1 and 2 will trigger and the PI3K will trigger to AKT activation of this protein after the phosphorylation lead to cause insulin resistance. So in this way the insulin will not bind with the receptor correctly, and in this way the GLUT channel will not open. The silymarin will also activate the GSH and SOD and CAT protein collectively. This will lead to inhibit the inflammation. Remember, and this is within the cell. These are protein which that in the cell. So for understanding, I am writing by this manner. So the SIRT1 gene activation lead to secrete the insulin, and in this way the insulin secretion more. But in the case of SIRT1 will inhibit the gluconeogenesis in the liver. The reactive oxygen species and FOXO reactive oxygen species will trigger the FOXO will inhibit the SIRT1, and in this way the gluconeogenesis ultimately inhibit. And in this way the silymarin will inhibit the reactive oxygen species and FOXO will inhibit, so the SIRT1 will trigger lead to insulin resistant inhibition. While on the other hand, the silymarin will also inhibit the apoptosis by the BCL2 gene trigger, while the BAX and caspase 3 will lead to inhibit the apoptosis. I hope you make sense about this is the benefit of the silymarin to create the anti-diabetic effect. Thanks for watching. See you in the next video. For this time, bye.